Thank you for Eugene's introduction, and it's my pleasure to share the audience of this meeting to introduce about our brand, Top Sales. And in this seminar, we are trying to introduce our proud product, which is a chemically defined cell supplement. And in this seminar, I will cross from the 50 years ago, the gold standard of cell culture, and until to the animal source material, and to the wonderland of the cell culture, which is chemically defined. And we also try to introduce that the top cells can provide the product and the different characters of the product, and what we can do to refine the issue you faced in every cell culture. So I am today's speaker, I am Sean. And in the beginning, I would like to introduce our company, Genesis Biomedical. Actually, we are Taiwan biotech, and most of our employee is a PhD or a master degree, and most of the employee is focused on the research and the development. And our business covered new drug development and also the proteomics reagent, which is under Vision 13, and the cell culture supplement and the cell culture tool, cell biology tool, which is under the top sales. So the top sales, we are trying to provide the top quality for cell culture. That's our brand. That's our layout we are want to do in this way. So about myself, about the speaker, I'm Sean, and I would like to share that my background actually is from the biochemistry. I major in the protein chemistry through the university study and graduate school study. And after graduate from school, I as a research assistant in Academia Sinica, which is the top public research center in Taiwan. And I also joined the National Taiwan University Hospital and to do the clinical sample study, the research. In that time, I was a lab manager of ENT department. So I am not only the salesperson. I have a biological and cell culture background, and I have a lot of failure experience. I mean, for the cell culture, for the contamination, for the incredible trouble post death of the cell, I have faced. So I cannot be a very successful researcher, but I can be a very good troubleshooter because I accumulate a lot incredible, unreliable trouble on cell culture. That's the reason why I understand the daily problem of cell culture workers, what they are working with, when they feel stressed because they failure cell culture. I can totally understand. And my publication is located from the early of this century and around 2010, and it's about a decade ago. So after 2012, I just become a traitor of the way lab researcher. So I go to the business development. I go to the BD salesperson. So let's take to today's topic, cell culture technology. So currently, actually, we are kind of to do the modern cell culture. I mean, it's around after 1960, 1980s. In that time, the basal media is really defined, and we have find the incubation condition, including the humidity and carbon dioxide concentration, and some trace element. And when we go to the 1990s, we started to discuss about that the potential risk of animal source of nutrition. So we started to focus on the serum-free culture. When we move to 2000, we start develop about the biopharma, I mean the biologic drug and the cell therapy. And right now we are focused on the organoid development 3D culture, and we are trying to using the iPSC or stem cell to do the gene editing and cell differentiation, and we will try to find a more safe and potency culture media, which is a chemically defined media. But currently, we do not achieve this goal right now. So mostly, we are using the animal source nutrition. So this is a basic culture media. Actually, we have already included some essential compound in the basal media. When we need to additional supplement, we will provide, for example, growth factor, serine, something like that. And the most important is that serine is kind of critical because it's a nutrition in the cell culture media. So in the past half century, the gold standard is animal serine. For example, fetal formula serine, FCS, something like that. But actually, this is the byproduct of the meat production. So actually, we do afraid that unknown pathogen. So it's a kind of we cannot see it, we do not identify, but we always afraid that something bacteria, virus in the animal we don't understand, we don't know. When we use this kind of cultured cell to injection to do the human treatment, do we have any threat or risk by the unknown pathogen? And the fourth point is that actually for this kind of the plasma, I mean the FPS is not intended to 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 provide to be culture level material. So it got lot to lot variance. So it's a very common experience through the whole laboratory is that we in the early of every year we need to purchase the whole year's lot of FPS. So we need the vendors to provide 3 different lots or more different lots of the product, and we try to find the best, the close to our previous lot one to do the whole year's purchase to prevent lot to lot variance. When we move to the serum-free serum, is that we are trying to avoid animal material. So this is a no animal. But instead of is that it's from the human. For example, the human serum is from the human every serum. The donor is from the healthy male and every group of blood type. And usually donate plasma to isolate human every serum. So you can imagine that the production, the lot size is very small. The cost is incredibly high. And in the early of 2010, we start develop a human fertilizer, which actually is for the human fertility. It's very easy. It's from the donated human fertility. When the fertility is expired, the manufacturer will collect from the expired fertility from the blood banker and to do freeze and thaw to release the alpha granule in the fertility. And inside there are a lot of cytokine growth factor is very helpful for the human cell culture. And another product, a common product is a human protein. For example, human albumin is a common nutrition source from the human to avoid animal material. But this kind of product, it has a limitation is that it only suitable for the human cell. For other mouse or rat cell, you need to be tested. Not every cell is suitable for human source series. So it's kind of a transient product. When you move from the animal source to chemically defined, is a transient product. Eventually, we think the chemically defined is the most safe, most clear, clear recipe. And in the cell culture field, in the cell culture area, we think that if you provide protein inside, but the protein, the purity and the identity is very clear. Even we contain protein inside, we will think it's chemically defined. And the most important that chemically defined provide a very stable lot-to-lot consistency. So when we try to have some reference, we search from the Merck website, and he just asked a question that why chemically defined is suitable for the bioprocess. So he recall our point of view that the first, try to reduce lot-to-lot variance and lower the risk of contamination. The unknown pathogen from the animal source, animal material, is a risk of contamination. So chemically defined, we can able to through the manufacturing to do by a GMP manufacturing process, we can clear and lower the risk. And when we need to do some PTM or differentiation, we are able to do this kind of ability attributes. So let's back to our top sales product. Currently, we have 3 different types of chemical defined cell supplement. And in our top sales, our more serious product, actually we are purely 100% chemically defined, which means we don't contain any protein, any cytokine, any recombinant protein inside. So we are purely chemical, which means we are very stable through the room temperature, through the shipment. We don't have in our internal data, we don't see any variance of the potency during the shipment or storage. Even our product, we label storage under 7 degrees Celsius. But as our testing, we put storage under the room temperature, 25 degrees Celsius, and avoid the sunlight direct to contact the product. We still can maintain the same potency and the quality of the product. So currently we have 3 different products. The first is a hybrid more. It's specific for the hybridoma culture supplement. Actually, this product is our senior, the first more serious product is specific for the hybridoma culture. And it has been our product line for more than 10 years. And the second one is Cytomore, which is for the cell rescue supplement. This product is aimed to try to improve the viability of the subculture cell. And we have a vision that we can apply on the primary cell, and we can reduce the serine cell culture. And the third part is the latest product is the neuron more, which is specific for the neuron cell culture. And compared to the other products on the market, we are the only product which is 100% clinical defined without any protein growth factor inside. And our cost effective compared to other products and have a better performance. So what is our functional ingredient inside? Actually, we are thinking that during the this reference in the 2005, we found that actually the energy threshold is very critical for the cell cycle. It's just like that. We have the energy threshold. If we can overcome, the cell cycle will go on, the cell will life. But if the energy level is too low, we cannot overcome the energy threshold. It will be a senescent, which is an elder cell, and without overcome the ATP level, it will stop in this cell cycle. Then eventually you will go to the death pathway. Eventually the cell will be dead. So energy inside the cell, the cellular energy is very critical for the cell. So that's the reason why it's very suitable for the cell culture. Just because cell culture is that we just want to make the cell able to do the proliferation. So during this product and this reference, actually we do have our internal testing through the by the more serious product. Young cell is easy overcome the ATP threshold is very easy because the young cell have a more energy, more good cell behavior. Senescent cell, the energy is too low, do not overcome, go to a cell death. But with our more serious product, you will increase the recover the ATP and close to the young cell one. Then eventually we can overcome and go to the next stage, cell cycle, and go to the cell proliferation. And the second part is that energy also included in the cell migration. So our more serious product, our functional ingredient, we are able to boost the cellular ATP. And the cellular ATP just like the battery and the fully charged battery inside the cell. So compared to the low ATP cells, fully charged, more energy, the migration will be better viability. So we have been tested in a different cell, and we found in our specific dose of cytomol, we can find the ATP level will increase 50%, around 20%, 40 to 60%. And we also test in the primary cell for the mice skin. You can also see that we have a significant increase ATP level compared to the vehicle group. So in the cytomol, we have another scenario application is that we can able for the primary isolation. Because most of some of primary isolation is by the semi-dry way. For example, we physical way to digest the tissue and attach on the culture plate. And we cover the, not fully cover, we just wait and contact with the medium with the digest tissue. And we allow the cell, primary cell from tissue debris to the culture plate. So in this moment, in this way, semi-dry isolation model, the cell migration ability become very important. So that's the reason why our cytomol is suitable for the primary isolation as a specific for the semi-dry way and accelerate the cell, primary cell from tissue to cell plate. So let's move to each product. First is hybrid mole. And as I mentioned, that is a specific for the hybridoma cultural supplement. And our goal is to try to increase the cell survival rate, especially for the hybridoma cell after cell fusion. And the idea is that the more life hybridoma cell during the HAT selection, the more chance, the higher susceptibility you can choose the right clone. So this is our design of this product. And this product has been in Taiwan, in Japan, in Korea for more than 10 years. And for whole three more serious product, we accumulate more reference paper, and we can provide after once you need. And we can also use it during the screening and screening hybridoma. For example, you have a selected right clone, and maybe you have already storage in the liquid nitrogen for a couple of years, and you need to diffusion, then you can use in a more. You can increase the viability after diffusion or in the subculture. So we have we compared to other way to culture hybridoma, for example, feeder layer or just regular FCS medium. You can find that the green one is a hybridoma is much better than the feeder layer and the FCS medium. And during the pandemic area, we have worked with several COVID-19 vaccine developer, and they are using the hybridoma compared with the feeder layer. So for the first reference is that they found that more post-fusion cell numbers compared to the feeder layer, and positive cell growth better with a hybridoma compared to other way. And in the other reference paper, we are compared with the PNH1, which is a very famous and common hybridoma cell culture supplement, which is the cytokine inside. And with comparison, is that we have a better signal to noise ratio during the ELISA screening. That's very easy because we are non-protein recipe, so we can reduce the noise ratio. And compared to the cost, we have excellent performance and cost-effective price. And when we move to our second product, which is launched in early 2020, and this product actually we do receive some customers that their complaint is we have some very rare or few researchers working with the cell, and they are very hard to culture, and we don't have other reference, other pioneer provide medium to do the cell culture, and struggle with the poor growth rate of the cell. So we are using our ingredient and to find a better way, refine the concentration. We do this kind of product, and it's specific for the cell culture supplement. So this product we don't have some species preference. We don't have the cell type preference. So for example, we can apply on the human skin fibroblast is kind of primary cell from the human, from the fibroblast. And then we can find in some, for example, dilution in 4 times, they can reach the higher performance. But for the bone marrow mesenchymal stem cell from the mouse, in the stem cell, and it's around 16-fold dilution is suitable, or 8 times is okay compared to the control one. We do have a very specific performance, good potency. And we also work with on the common carcinoma cell line. And for this design is for confirm our scenario to reduce serum use. For this design is that sometimes some experiment design we need to do the second level of subculture under the low serum or non-serum condition. For example, if you want to recover exosome from the cell culture, so we are trying to avoid the serum sourced exosome to contaminate of the cell one. So we need to reduce our non-series condition. So when we do 2%, is only 20% of original one with cytomol and the 2% FPS. We compare to 10% original concentration. You can find that after 4 days culture, 2% FPS with cytomol, the cell number, the cell density, almost the same compared to a 10% FPS. 2% FPS is good, but not so good. The numbers is reduced. It's not so many compared to the control one. So this is confirm our ideas that we can reduce the serum usage during your cell culture. I always told my customers that cytomol is kind of a Red Bull. You can give you instant energy, but it cannot replace the nutrition. So for the long-term culture, you just put a cytomol inside. Eventually, you don't have a very good result because it is not a nutrition. It's a Red Bull and non-sugar Red Bull. Yeah. But sometimes when I speak with Red Bull, some customer concern that this is coffee? No, of course not. It's not a real Red Bull. It's just my way to demonstrate it's an energy booster. Okay. And the second one actually is a bone marrow kidney cell, which is a very difficult culture and defined from my customers. And we are using 20% host serum. Then we compare with the 10% host serum with cytomol. I know what you think about the results still suck, but compared to the original one, yes, with cytomol reduced 50% usage, we maintain almost the same. But the 10% we can foresee the result is the cell become fewer and fewer, eventually nothing inside. The cell go to the death pathway. Yeah. So the third product is our neuromore, which is enhanced cell proliferation. So we have been tested in the N2A, which is a mouse cell line. Another is the SMA-A9, which is also the mouse neuron cell line. And in the different products, we can find that a different cell type. Some is increased on over 100%, another is 35%. And for this result, the second part we need to consider is that this product is able to change any city marker morphology and the potential differentiation or the cell ability. So in the PQP, we can find the morphology is almost the same. The cell type, the cell shape, the cell accumulation condition is just like the wild type one. And we do have the city marker staining, the separation, and the density, the level is the same with the wild type. And here is my friendly customers. He tried to do his test. He is working with the animal neuron stem cell. So this test just released 3 different categories. The first is the primary cell. The second is the neuron cell. The third part is the stem cell. So for the neural stem cell, the most important to show the stainless, I mean the ability for differentiation is about to formation of a neural sphere. So you can find that when we culture 7 days, this is control one, this is with a neuron wall 7 days. When we zoom in, you can find the neural sphere is formation round and very nicely. And we also try to test the city markers, still maintain the differentiation city markers on it. And we also have an additional application on the iPSC because this customer he is working with iPSC-derived neuron cell. for the mouse. So before they proceed their experiment, they are using the neural mole to do the iPSC culture. So you can find that in the higher concentration, in the original one, neuron mole concentration, we suggest the result is the best compared to a control one. And then more importantly, even in a whole different dilution, different concentration, the morphology is still the same with the original iPSC and do not show any unexpected differentiation result. So this is our goal. We only focus on the increase of viability, increase the proliferation rate. We do not change the original cell behavior just because we just increase the energy inside the cell. So in here, we just do the application table for you are easy to choose which kind of more serious product you should work with. For the first is epithelial cell. We have been tested in the human epithelial keratinocyte, which is cell line. We have been tested in human and mouse primary skin keratinocyte, dermal papillary cell from human, mouse, and rat, and the oral corneal epithelial cell from human and mouse. In this kind of category species, we suggest use cytomol. And for the fibroblast, from the primary one or from the cell line, from human or from mouse, cytomol definitely. Stem cell, mesenchymal stem cell, iPSC, or the cerebral gland cell, which is a polypotent cell to produce cellularly. From human and mouse, using the cytomol. And the neuron cell, from mouse, rat, primary neuron stem cell, iPSC-derived neuron progenitor cell, all related to the neurons, just use neuron mole. And for the antibody protein production, for example, for the HEK293T, which is a transient overexpression in human, suggest in cytomol, chill cell to do the protein production for biologics pharmaceutical. We suggest using the cytomol. Hybridoma for the hybridoma cell for the protein antibody production, definitely using hybridoma. And the other specific, unique, strange, hopeless cell culture, you can just try cytomol. For example, we have a professor, they are working with the European eel. So he can use this to culture primary muscle cell. And another very interesting story is a culture with a spider. They are working with a silken gland cell, and which is a primary cell. And they have been tested different kind of serum and supplement. Eventually, they successful culture under the cytomol. So if you are working category, we are working cell on this table, you can reference our table. If your working cell do not include in this table, please contact with our Korean distributor and share us your information. We will provide the proper concentration and a proper product for your testing. And under top cells, we also have other cell counting tools. For example, NTT assay and the CCK8 assay. And actually, our CCK8 will provide a powder form, which is stable and easy to work with. And actually, the quality and the performance we can compare to other have been known brand on the market. but we have the cost advantage price. And we are arranging upcoming products. The first is for a cryopreservation solution, which is the animal components free, chemically defined, ready to use, and contains DMSO, but easy to storage, and only 2 to 8 degrees Celsius. We don't have a 2 to freezing required, which is able to suitable for minus 80 degrees cryopreservation. If you short time, we do not able to put into the liquid nitrogen. And all kind of cell type and a research grade available and a GMP grade will be provided on your demand. So here is our free-home message that in the top sales in the most serious product, we provide 100% clinically defined cell supplement. Easy to storage and easy to operate. We don't worry about the loss of efficacy, potency during the failure shipment. And for our latest product, Neuromore, we are good on the neural cell proliferation with the neural marker expression and no more morphology. So thank you for your listening for this seminar. And I do understand if your culture cell not happy, your life will be not happy. So if you have trouble with your working cell, please let us help you nursing your culture cell with a more serious product. So please join our trial program, and we will provide a whole bottle and a standalone product for your testing. And for this slide, how about Shirley, you do the following introduction.
Yes, thank you for the webinar presentation from Sean of Anagenesis. For the Q&A session and the trial program will be held by the hosted today from the Philly, Korea.